Hello and welcome to Ph06 video lessons. Today we are going to be talking about challenge and response, which is one of the most important ways of our body defending ourselves. It's the way that we go for a specific response against a specific disease or a specific antigen. So, the idea of challenge and response is that when a pathogen enters our body or tries to invade our body is going to be fighting back. So the first thing that happens is that anything that's foreign, any antigen, any protein, the membrane of a pathogen or the protein of a virus or whatever, anything that doesn't belong to our body might be identified as an enemy and is going to be triggering this entire response that's going to be including the production of antibodies. So we have a pathogen here. In this one, I'm depicting a bacteria with the tiny little red antigens. These antigens can be a protein in the membrane, they can be a protein of a capsule of a virus, they can be a glycoprotein, they can be a glycolipid or whatever. It's something there that can be identified as enemy. This antigen, therefore, is going to be identified by a cell, the phagocytic leukocyte or the macrophage, the big eater, that's going to be looking and say, hey, you don't belong here. Every time a macrophage sees one of these, say, you don't belong here, so your food, I have to destroy you. So what they are going to be doing is that they are going to be phagocytizing it, is going to be eating it, engulfing it, going around, closing it, and voila, now the pathogen was eaten. It's engulfed inside a vesicle, and the macrophage is going to be destroying it. A vesicle with digestion enzymes called lysozymes is going to be put together with the vesicle containing the pathogen, and the pathogen is going to be destroyed, digested, and now is going to be remaining only pieces of it. And the pathogen was destroyed. However, the antigens, they may be kept. The macrophage, when they destroy the pathogen, they may keep the antigens to be able to show outside of its cells in a special composition of its own membrane protein and the antigen, which is called MHC. When the pathogen is together with this protein, it's not going to be identified as an antigen by itself, but actually because of this guy, this is going to be identified as, hey, look what I found. This macrophage now has the capacity of going around and advertising, I found an enemy, this is what you're looking for, let's destroy this guy. It's the equivalent of soldiers that in a castle invasion, they find a soldier of the other army. They cut the head and they show the helmet, say, hey, this is what you're looking for. They show for other cells what is the enemy to start a defense, a response to be able to destroy this enemy that are going to come in a very big number. Okay? So, the macrophage, they eat the pathogens. Hey, so what's the problem? How do we get sick? Macrophages, phagocytic leukocytes, they kill pathogens, but they don't kill them fast enough. No, we have some of them, but they don't manage to kill every single pathogen that are growing, multiplying, so they need help. So in this case here, after eating the pathogen and presenting the phagocytic leukocytes, they are going to be looking for T cells, which are lymphocyte T or leukocyte T, that are going to be capable of reacting to this antigen. So they will look around, and once they find one that is capable of working with this antigen here, they activate this T cell. So now you have an activated T cell. Hey, I am also looking for this guy. So this T cell now is going to start its job looking for this antigen and carry on the response to be able to fight for this this war against this antigen. So what's going to be happening with the T cell? It's going to be copying itself. It's going to make copies and clones of itself. Some of them are going to be called T helper because they're going to be helping in the remaining of the response. Some of them are going to be called T cytotoxic or T killer. They are going to be doing another job in fighting the disease. And some of them are going to be called T memory. They are going to be memory cells. This T memory, by the way, they are activated cells against this pathogen that are kept aside because next time you get the same antigen entering your body, we don't need to go through all the process of eating and then activating and then, no, we already have an army of the memory cells, the T memory cells, to be able to fight the disease in a faster way. Okay? So this here is what happens to the T cell. So now let's put these different types of T cells at work. The T memory, as I told you, they are held back. We keep some of them, some thousands or maybe some, no, many thousands of them, which is a small number. They are held back for the future. Just in case next time you have an invasion, they start the reaction much quicker. Infected cells of your own body, they have a problem. It's very difficult for them to kill themselves, to cure themselves. So what the T cytotoxic is going to be doing is that they're going to be looking for the cells that have been infected and they say, there's no cure for you. Unfortunately, you are contaminated and it's not possible to cure you. You are like a zombie that the only way of getting rid of a zombie is killing the zombie. So I'm sorry, cell, I know you're part of this body, but unfortunately, I can't cure you, so you have to die. So they say, would you mind dying? Would you mind killing yourself? And the cell says, okay, and they kill themselves. Our cells are very obedient, usually. And when they're told that they have been infected and that they have to kill themselves, they just go there and they kill themselves. And in this process, they destroy the entire army of pathogens that are hiding inside of them. Yay, very good. So the T cytotoxic is very good to do this part of the response, killing the infected cells of our body. What about the T helpers? T helpers, they are going to be helping to activate B cells. They are going to be looking for B cells that also react to this same antigen here, the one that we found, this little red ball, and they are going to be activating it. And once activated, the B cell are going to be cloning itself and is going to be making many memory cells. So again, we have memory B cells that are going to be kept aside for future invasion, so we can respond much faster for a future invasion of the same pathogen. And they are going to be making many clones of themselves. They are cells that are capable of producing antibodies against this antigen. And now we are going to have an army of plasma cells, that's the name of the B cell that have copied themselves, and are starting to produce antibodies. So the plasma cells produce antibody, many of them, and they bust open and they release a lot of antibodies into our bloodstream, into our body. And these antibodies, they are like guided missiles that are going to be destroying the pathogen. So this here is the process of challenge and response. And the idea of antibodies is this. For example, antibodies, they can glue around viruses and bacteria, neutralizing them. So they attach to the virus protein capsule. So the proteins, they cannot do the job, or they attach to protein in the membrane of bacteria and they cannot do the job. So basically, they are going to be neutralizing the pathogen. They can also agglutinate pathogens. They glue one, then glue to another one, and then to another one, and they glue many of them. So in this case, on a macrophage, it's one. It doesn't eat one. They can eat dozens of them at the same time. Antibodies, they can also glue to dissolved antigens. So, for example, snake poison is a dissolved protein in your body that creates havoc. The antibodies go there, they glue to them, and once they have been glued, they are not dissolved anymore, so they precipitate, and then the macrophages, they go there and they can do the job and they can eat them. Okay, there are other ways that the antibodies are going to be working, but those here are the three main ways for antibodies to attack. They are very, very, very specific to a pathogen, very efficient, and once you produce them, the pathogen don't have a chance. So this is the way. I will put here everything going again, just narrating for you. Macrophage have eaten, activate T cells copy themselves, make T helper, T killers, make T memory cells for the future. The T memory are held back, okay, they are over there. The T cytotoxic, they are going to be telling the infected cells there's no cure for you, would you mind dying? They kill themselves, so you destroy the ones that have been infected. The T helpers activated the B cells that are going to be making memory cells, but also going to be making plasma cells that are going to be releasing a lot of antibodies. And then you have a response and you are cured, yay, and this is the idea. We have the potential to produce antibodies against any antigen in the universe. If there is a virus in Alpha Centauri that humans have never even heard about that it could exist, 4 light years away, and once we get there, when you get in contact with that guy, we will have T cells and B cells that supposedly would be capable of producing a response to produce antibody to kill, to destroy those antigens in Alpha Centauri. So theoretically, we could cure ourselves against any antigen, or to kill any pathogen, destroy any pathogen in the universe. The point just is how fast we can do that. If we do that fast, we don't get sick or much sick. If we do that slowly, we can get sick or very sick, and sometimes we don't manage to kill them fast enough to save ourselves. But potentially, we have the potential to destroy any antigen, to destroy any pathogen. Okay. Just to show the process, how things goes over here, macrophage, activating T cell, B cell, so all the stuff that you saw before. And now I have a quick quiz here for you. I hope you have learned about this way of very specific defense. This is the way that we get ourselves cured most of the time by doing a specific response to an antigen. Do the quiz and I'll see you in class. Bye bye.